So this chapter is called The Working Cell. And in this chapter, we're going to be looking at membrane structure and function, the energy of the cell, and how the enzymes function in the cell. So the first thing is the membranes. Membranes are very important because they are what separates the cell from what's inside the cell from what's outside the cell. So every cell has a plasma membrane. And we have a representation of the plasma membrane over to your left here. The plasma membrane consists of a phospholipid bilayer. So this is the phospho part of the phospho, and this is the lipid. The head here is hydrophilic and likes water. And the tails, you'll see the tails all go together. I'm drawing them in, and those are hydrophobic, and they don't like water. Embedded in that lipid in the bilayer, we have different proteins. We've got an alpha helical protein, we've got a globular protein, we've got channel proteins that allow molecules of interest to cross through the membrane. And we also have glycoproteins, these green things on the outside that help cells to recognize each other. I guess that's supposed to be a glycolipid. This is supposed to be a glycoprotein. But anyway, there are sugar molecules on the outside. So this model is called the fluid mosaic model. And it's called the fluid mosaic model because these proteins can be seen as being suspended or floating in a fluid phospholipid bilayer. So the proteins will bob around and move around within the membrane. So plasma membranes exhibit selective permeability. This just means they are permeable to some things. Some molecules can get through, and other molecules are restricted from entering the cell. So very small molecules like oxygen and carbon dioxide can diffuse across this phospholipid bilayer, this hydrophobic interior, but some things can't. Phospholipids will spontaneously self-assemble into simple membranes. And this is important when we start talking about how life began, because one of the first requirements is going to be to have the self separated from what's outside the self. And so the fact that phospholipids will spontaneously form a lipid bilayer like this means that the very first stages of life, one of the first requirements, can be met. So I've said that the membrane here is selectively permeable. So now we need to talk about transport across it, because we do need to get things into the cell and things out of the cell. Some movement across the cell membrane requires no work, and we call this passive transport. Substances will move from greater concentration to a lesser concentrated. So if I open a bottle of perfume in the front of the room, some perfume molecules will come out of the bottle. That's my bottle. Like I said, I'm not in art school. And they will move, these concentrated molecules will move to where they're less concentrated. So if you're standing in the back of the room, you may not smell the perfume at the beginning, but maybe by the end of class you would. So let's look at diffusion in action. So here we've got green molecule. We want to show concentrations. That green bar shows how many of the little green molecules are on the top. And we have a membrane in between. This yellow thing is the membrane. We're going to put some channels in so that the green can cross the membrane. This would be a passive diffusion. The cell would not need to have any energy expended to complete this. And as you'll see, initially, more molecules will move down than move up because there's a greater concentration of the green balls on the top. Eventually, we'll get, in fact, I think it's already happened, we have about the same amount of green on the top as we do on the bottom. And we've reached equilibrium. So let's see how this works in the cell. There we go. Substances always travel down their concentration. Their concentration gradient in passive transport. So they're always going to go from a greater concentration to a less concentration. So simple diffusion. In simple diffusion, no energy is required. And it's the movement of a substance from an area of high concentration to an area of low concentration. Examples would be oxygen diffuses through the membrane, carbon dioxide diffuses through the membrane, and other very small molecules. Facilitated diffusion also requires no energy. It's the diffusion of a substance down its concentration gradient through a membrane through a transmembranous protein channel. And I've got a picture of that, of a kind of a cartoon of a channel down here below. Molecules that use a transmembranous protein channel would be things like glucose and polar charged molecules. The charged polar molecules can't get past that hydrophobic layer in between, in the middle of that membrane. Can't write and talk at the same time. All right, down here we've got a cartoon of a protein channel. And so what's happening here is there are more of the little balls on the outside, and so some of them will just pass through. This channel does not influence the direction of flow, and the balls could flow out the other direction as well. But as you saw in the thing I showed you, the simulation, if there are more of the little balls on the outside, they're more likely to flow this way. And so the net flow will be from greater concentration to less concentration. Now, facilitated diffusion requires a specific transport protein. So this protein that's here is going to be for only one molecule, only one type. So this might be a glucose transport. So it would allow glucose to traverse the membrane, but it wouldn't let anything else. They're very specific. Only let the one thing through. So water can diffuse across a semipermeable membrane. And this has a special name. It's osmosis. Diffusion of water across a semipermeable membrane has its own name. It's osmosis. So let's look at osmosis. Look at the U-shaped tube. Let's first look at the one on the left. And you'll notice that on one side we have a lower concentration of the solute. That's these little purple things. So there are fewer little purple molecules on the left side of this left tube. In the middle, we've got a semipermeable membrane. So water can pass through this membrane. But the little purple solutes can't. So that gives us that there's more water on the right side than on the left side, because some of the space on the left side of this U is taken up by having more of these little purple things. Okay, so water will move from the right side to the left side so that you'll to give more equal concentrations of the solute. Of course, water doesn't think about doing anything, but I like to think that water is trying to dilute the more concentrated side. So it will move toward the more concentrated side. Now, water can pass through a membrane using an aquaporin. And this is a specific channel, just like the channels we were talking about moments ago up here. We talked about facilitated diffusion. This is a way to facilitate diffusion through the membrane. And what this diagram over here is supposed to be showing you is this is water, and this is water on this side. The light yellow or gold balls, that's the phospholipid. And then the green are the hydrophobic tails. So over here we have other, I'm not going to draw this I needed to put longer tails on the other side, but anyway. And what you have here in the middle that's depicted in blue is a channel that's allowing water to pass through. And you could see the little water molecules as they're passing through the membrane on in that little channel. So that's a facilitated diffusion. The water is just diffusing. One more thing, I've got a simulation to show you of osmosis. So here we've got a membrane that's this middle line here. On one side we have pure water, and on the other side we have water and sodium ions. Notice that the sodium ions are each surrounded by a lot of water, and they're too big to go through the membrane. So only the water can diffuse. That's why this is osmosis. I'm going to reset it, and when I reset it, you'll see that all the free water, all the water that's not bound to the sodium ions, will be on the left side, and on the right side we'll just have sodium and water bound to it. So let's see what happens. All right, so first you see that water is going through the membrane. The sodium can't go in through the membrane, so it stays on its side. And it's like the water is trying to dilute the sodium ions. Eventually, the amount of free water on each side will be about the same. There are 30 water molecules bound to the sodium. So it looks like one side has 22 and the other has 20 free waters. So let's look at the 3 examples I've made here. Each of the black circles is a cell, very simple cell, but a bilayer membrane that's water permeable, but my large green molecule can't pass. So let me get my green molecule here. In the first one, we're going to have green molecules on the outside, but we also have them on the inside. And this is going to be roughly the same concentration. So the water molecules are able to cross the barrier, and what we'll find is that some of the water molecules will diffuse across out of the cell, but it's just as likely that a water molecule will diffuse in. So this solution has the same concentration, and we call it isotonic. And the cell maintains its shape. The cell does not change. In our second example, we're going to put it in a solution. Let's see if we get the green back. We're going to put the large solute molecule on the inside. Now the green molecule can't pass the barrier, but the water can diffuse normally, and the water will diffuse in. And this will result in the cell bursting open or lysing. This solution is hypotonic. You can think of the solution as being low, and that O, hypo, is low concentration. This last cell is going to have our large green molecule on the outside, but none of it on the inside. Well, the water that's inside is going to be drawn out. It's going to diffuse out to try and dilute out the green, and this will cause the cell to shrivel. I've got these drawn below. Okay, and this solution we say is hypertonic. I'm going to write it down here. And hyper means high. If you're hyper, then you have too much energy. And so a hypertonic solution has too much solid. The same root word. Okay, so let's fill in our table. 2 solutions that have the same solute concentrations are isotonic. In a plant cell, this will make the plant cell flaccid. And it'll make this is a red blood cell, it'll make it normal. We've got as much water going in as we've got going out, so that makes it not change. Hypotonic has less or lower amount of solvent, and it makes the plant cell turgid. because what we have is water going in, trying to dilute out all that solute. And it will cause a red blood cell to lyse. Whenever you give blood or fluids to a patient in an IV, you have to make sure that it's an isotonic solution. Hypertonic is a solution that has too much or more solute, hyper, and it will cause a plant cell to shrivel. And they'll do the same thing to a red blood cell. It does like that. Okay, so we've talked about passive diffusion and osmosis, which is also passive. Not all movement across membranes is passive. Some of it requires work from a cell, and this is active transport. And an energy molecule, usually an ATP, supplies the energy. An active transport will pump a substance up its gradient. So it'll take it from where it's less concentrated to where it's more concentrated. And we have a cartoon of active transport. The green molecule binds, and then ATP is used, and then it opens on the other side. So we're pushing the green ones this way. Okay. Well, there's some molecules that are too large to be carried on protein transport, and these materials have to be packaged into a vesicle so that they can be moved up their transportation, moved into the cell. So this is called exocytosis. And that's the movement of things going out. So it's exiting. If the stuff is exiting, it's exocytosis. Endocytosis is things going in. And we're going to look at two different types of endocytosis. We'll look at phagocytosis, which is the engulfment of a particle by the cell wrapping the membrane around, forming a vacuole. And we have a picture of that here. The cell just wraps itself around this and makes this vesicle. The other is a receptor-mediated endocytosis. In receptor-mediated cytosis, there are these receptors, these Y things here, these red Y things are the receptors, and they are holding on to a molecule that's of particular interest, these purple triangles. And they grab onto the molecule, and then the phagocytosis occurs, and the endocytosis occurs, and you make a vesicle. And so it's to pick up a particular solute. You want to pick up a particular molecule. Okay.